Hello everyone, welcome again to the series of solved previous year's ICAR PG examination, Plant Science, where we will discuss the question papers of Genetics and Plant Breeding. This time we will discuss 2018 paper. Question number 1, EDVs are, so they are called essentially derived varieties. These are the varieties which have been essentially derived from existing variety by the means of genetic engineering, mutation, or tissue culture technology. Question number 2. Tifton A cytoplasm is found in? It is found in pearl millet. Cytoplasmic male sterility. It was observed in pearl millet in Tifton city of Georgia. Question number 3. In terms of ploidy, trisomy is a form of? It is a form of hyperploidy, that is 2n plus 1. What happens is the chromosome number is greater than the normal diploid, for example Down syndrome, which follows trisomy. In case of hypoploid, here the chromosome is less than the normal one, for example Turner syndrome. Question number 4. In which direction do the F3 phosphodiester bonds run in Watson and Crick DNA model? So they run in the anti-parallel direction, which makes the DNA stable. Question number 5. The genetic gain under selection is more rapid if? The correct answer is option D. The heritability should be 100%. Genetic gain is the difference between mean phenotypic value of progeny of selected plants and the original parental population. That is, the genetic gain is equal to xp minus x0. Question number 6. According to the dominance hypothesis of heterosis, which one of the following is a false statement? The false statement is option B. Inbreeding depression is due to the harmful effects of dominant alleles. Instead, it is due to the homozygosity of deleterious recessive alleles. The term dominance hypothesis, it was given by Davenport. Question number 7. The unit of radiation dose is called Gray, and 1 Gray equals to? It is equal to 1 joules per kg of the irradiated object. Also, there is a relation, that is 1 Gray. It is equal to 1 joules per kg, which is equal to 100 rads. That is the dose of radiation. That is again equal to 10 raised to power 4 erg per gram. Question number 8. Total number of nuclei involved in double fertilization in normal diploid plant is? The correct answer is option C, 5 nuclei. As we can see in syngamy, one male gamete fuses with the egg cell, resulting in the diploid zygote. Also, in case of triple fusion, the other male gamete fuses with two polar nuclei, resulting in triploid endosperm nucleus, that is 3n. This implies 5 nuclei are involved. Question number 9. How many types of gametes will be formed in F1 in a trihybrid cross? Trihybrid cross means n is equal to 3, and therefore the number of gametes should be 2 raised to power n. This implies 2 raised to power 3 is 8. The correct answer is option C, that is 8. Question number 10. In sterility of the plant is determined by the result of mutation in mitochondrial genome in cytoplasm, then it is called, so it is called cytoplasmic male sterility. Question number 11. Single nucleotide polymorphism is? It is biallelic, that is 2 allelic variants they are observed. So SNP or single nucleotide polymorphism, it is a condition where a DNA sequence variation is seen when a single nucleotide is altered in the genome. Question number 12. Heterostyly is observed in? It is seen in linseed. Heterostyly is a condition of having styles of different lengths with respect to the stamens. In case of hercogamy, the pollen grains fail to reach the stigma. For example, lima beans. Question number 13. In recombinant DNA technology, which enzyme is used to join fragments that have been cut by restriction enzymes? The correct answer is option D, DNA ligase. Question number 14. The hybrid progeny between a single cross and an inbred line is referred to as, three-way cross hybrid. The inbreds I1, I2 and I3 are taken. I1, I2 they are crossed. The F1 produced is then crossed with I3. Question number 15. In a crop plant, if the percentage of cross-pollination exceeds 5% and may reach up to 30%, then the pollination behavior of such crop species is referred as, the correct answer is option C, often cross-pollinated. If it is more than 30%, then we call cross-pollination, and if it is less than 5%, then self-pollinated crops. Question number 16. In tRNA molecule, the D arm is named for the presence of the base. The base is called dihydrouridine. The D arm is a feature of tertiary structure of transfer RNA. The main function of D loop is of recognition and it contains the base dihydrouridine. Which of the following amino acids are aromatic in nature? The correct answer is option number D, phenylalanine and tyrosine. Question number 18. Heterobeltiosis is the superiority of F1 over. It is over beta parents. The term heterobeltiosis, it was coined by Blitzer. It has the formula F1 minus the beta parent over beta parent into 100. Question number 19. Bioassay of cytokinin is done by conducting. It is done by conducting the cotyledon expansion test. The dwarf maze test is done for the Gibberella and Avena coleoptile is for auxin. Question number 20. If total number of n inbred lines are involved, then the total number of possible synthetics is, the correct answer is option D, n is equal to 2 raised to power n minus 1. Question number 21. The enzyme GOGAT stands for, it stands for glutamine to oxoglutarate aminotransferase. This enzyme has a role in synthesis of amino acids and proteins. Question number 22. The recent molecular technology which is used to determine the exact order on the basis of genome is, it is called DNA sequencing technique. Which of the following represents genomes and chromosome number of the oilseed Brassica napus, subspecies napus? The correct answer is option A, AACC and 38. As we can see, Brassica napus is a cross of Brassica rapa and Brassica oleracea, which gives n is equal to 19. Therefore, 2n is equal to 38. Question number 24. The process of breeding food crops that are rich in bioavailable micronutrients is referred to as, it is called biofortification. Question number 25. Who proposed the concept of gene pool? It was proposed by Harlan and DeWitt in 1971. It says there are 3 gene pools, that is primary, secondary and tertiary. Question number 26. If the sequence of bases in a section of DNA is TAGGC, TAA, what are the corresponding sequenced bases in mRNA? So in mRNA, thymine will be replaced by uracil. The correct answer is option C, AUCCGAUU, where the thymine is based with A and adenine is paired with uracil. Question number 27. XYZ system for hybrid production was proposed for, it was proposed for wheat and the term was given by Driscoll. It involves 3 lines, that is XYZ. Question number 28. A set of germplasm accession derived from base collection to represent the genetic diversity in the whole collection is called, it is known as core collection. Question number 29. The viable gametes produced by a translocation heterozygote is in, it is in alternate segregation. Here the 2 non-homologous chromosomes and 2 translocational chromosomes, they separate into different poles. In case of adjacent 1 segregation, homologous centromeres pass to opposite poles and in adjacent 2, they pass to the same poles. Question number 30. What is the source of D-genome of wheat? It is from Aegilops toschi. Question number 31. While analyzing the structural variation in DNA, the B form of DNA consists of how many base pairs per helical turn? The correct answer is option number B, 10.5. Here we can see the A DNA contains 11 base pairs, B DNA has 10.5, whereas Z DNA has 12 base pairs. These are some other characteristics of these three DNAs. Question number 32. Minimum population size required for recombination of all F1 gametes at random is? The correct answer is option B, 3 raised to the power n. If we see for gametes, it is 2 raised to the power n because there are 2 gametes. If we say for genotypic ratio, we calculate the population size at 3 raised to the power n as we have 3 different genotypes. In case of phenotypic, we see 2 raised to the power n because we have 2 phenotypes. The complete population is measured by 4 raised to the power n. Question number 33. Assuming the Hardy Weinberg equilibrium, if the frequency of 2 alleles at the gene being studied are 0.6 and 0.4, the genotypic frequency of heterozygotes will be. So if we see, the equation is p square plus 2pq plus q square. We have been given p is equal to 0.6 and q is 0.4. So we have to calculate the frequency of heterozygotes. That is 2 into 0.6 into 0.4, which will 0.48. Question number 34. Okazaki fragments are related to. So they are related to the DNA synthesis. The Okazaki fragments, they are short lengths of DNA that are produced by the discontinuous replication of the lagging strand. Question number 35. The multiple replicates of same chromosomes hold together in a parallel fashion is called. So it is known as polyteny. In case of endomitosis, the replication of chromosomes in the absence of cell or nucleodivision results in the numerous copies of each cell. Whereas in the polyteny, we have huge chromosome puffs which are known as Balbiani rings. Question number 36. While explaining cell organelles in ribosomes, the suffix s in ribosomes unit indicates sedimentation coefficient. Question number 37. Entry of the pollen tube through micropyle is known as porogamy. If the pollen tube enters through the chalazal end, it is known as chalazogamy, and if it enters through the integuments, it is known as mesogamy. Question number 38. The enzyme reverse transcriptase was discovered by Temin and Baltimore in the year 1970. In a type of apomixis known as adventive embryony, the embryos develop from the nucellus and the integuments. Question number 40. A cell contains 40% chromatids at the beginning of meiosis. After the completion of meiosis, the cell would contain. So we see that 2 chromatids, they lead to 1 chromosome. This implies 40 chromatids will give rise to 20 chromosomes. And the meiosis, here the reduction division occurs. So haploid number will be 10. That is, the answer is 10 chromosomes. Question number 41. If this mRNA is translated into protein, how many amino acids would it contain? Here we can see UAA, it is a stop codon. So this implies there will be 4 amino acids. If a MS line is crossed with a heterozygous male fertile plant and the progeny is segregated as 1 male fertile and 1 male sterile plant respectively, then the male sterile is termed as genetic male sterility. Question number 43. Apomictic seed is a seed produced by, without fertilization. Question number 44. Multi-parent advanced generation intercross population, that is MAGIC, generally requires how many members in crossing? It requires 8 parents. So these populations are large sets of recombinant inbred lines that are on a genetic mosaic of multiple founder parents. Identify the best suited stage of cell division for in situ hybridization in the species where chromosome length is very short, for example pulses. So the correct answer would be Pectine stage. Question number 46. The phenomenon of Wobble hypothesis does not comply with which of one of the following? So it does not comply with the answer that fewer mRNA molecules than expected. Rather, it explains the genetic codes, they are degenerate. Question number 47. Triticale is a man-made cereal produced by the crossing of, so the correct answer is option A, Triticum aestivum and Secale cereal. Which variety of pigeon pea was used for whole genome sequences? It was Asha. Question number 49. The cell organelles that act as factories which translate the genetic information from nucleus to usable protein molecule is, so they are known as ribosomes. Question number 50. The first enzyme to be purified and crystallized was, it was urease. The apomixis that occurs commonly in citrus species is, it is known as sporophytic apomixis. Here the embryos develop from somatic nuclear cells. Question number 52. 7 characters of Mendel's experiment were located on how many chromosomes? They were located on 4 chromosomes, that is 1, 4, 5 and 7. The classical work of Beadle and Tatum on Neurospora crassa has proved that, so they have proved that one gene is responsible for synthesis of one enzyme, which is known as one gene, one enzyme hypothesis. The tendency of genes to remain together in the same gametes during segregation and not show independent segregation is found in, it is found in linked genes. Question number 55. In a self-pollinated variety, if a mechanical mixture was caused during threshing, then which breeding method would you adopt to purify the variety? For purification, we go for pure line selection. The 3 nucleotide combination adenine-uracil and guanine, they code for methionine, that is also known as the start codon. Question number 57. Histone proteins found in the nucleus are rich in, they are rich in lysine and arginine. Question number 58. In butterflies and moths, the sex determination system is, the correct answer is option C, ZW and ZZ, where we have a heterogametic female and a homogametic male. So that was it. Thank you.